First off, there's a few people I'd like to thank. Cindy Stein and Claire Putman for helping with basically everything. Matt Newman found my article. Mark Strockman helped me in the early stages, and so did Manicelli. Jackson Hoyt has helped me with a lot of miscellaneous things. And obviously Mr. D, the ever helpful Mr. D, and the Journal Club team. So my article is a gene-specific role for the SSU72 RNAP2 CTD phosphatase in HIV-1 TAT transactivation, or protein in HIV hijacks human DNA to create more HIV. So before we begin, a little bit of background on HIV is needed. For example, for starters, HIV is a retrovirus, which I'll explain in the next slide. Without treatment, there's a 9 to 11 year survival time, and it transfers through bodily fluids. And it's really important because a lot of our parents still remember when HIV started. It's recently discovered, it has a long history, and it's still fresh. So what a retrovirus is like other viruses, that's a retrovirus. It has RNA, but it takes that RNA, it uses reverse transcriptase to turn it into DNA, and then integrates it into the host's own DNA. So a person infected with HIV has HIV DNA in their own DNA. And when their cells replicate, HIV replicates. After retroviral DNA is introduced into, it's integrated into the cell, it's normal transcription, normal translation, DNA, RNA, proteins. So the protein that the article looked at mostly was TAT. And what TAT is it enhances HIV transcription. So it causes more HIV to be made within the cell. And it's already a well-established protein. It's known to have a lot of interactions with human proteins and other viral proteins and whatnot. But this article found a new link to a protein that was previously not thought to interact with. So what TAT does is it works on RNAP2, which is RNA polymerase II. RNA polymerase II takes DNA and transcribes it into RNA. Basically, TAT is connecting to all of these proteins and working on the CTD, which is just the tail at the end, and it's changing various configurations within it. And RNAP is affected by CTD phosphatases, which becomes important here. SSU72 is the protein they found a link to. It's a CTD phosphatase, which means it removes phosphate groups from amino acids in the CTD, in the little tail at the end of RNAP2. And first, the researchers looked at finding a link between TAT and SSU72. So they just did a protein analysis, and what they found was in TAT positive cells, there was SSU72 activity. TAT negative cells, there was no activity. And over time, there was increase of activity. That's important because it sets up the rest of the article and finds this new link.
Is there any particular reason you chose to use HeLa cells in the first figure?
No, just because they're common. So what SSU72 normally does is it, like I said, it removes phosphates from amino acids in the CTD. Specifically, it works on S5P and S7P, mostly S5P. And what TAT does is it was found to basically move this process along. So as you can see, as they increase levels of SSU72 with TAT positive, S5P and S7P activity decreased significantly. So basically, they found that there was a link between TAT and SSU72's activity being increased.
What's GST?
GST I think it's another TAT that goes along with the mutated SSU72, but it's unrelated to it. So basically, they hypothesized that SSU72 is essential to TAT function. So here in this graph, they look at what happens when they increase SSU72 levels and TAT levels. What they found was that as they increased together, so did production of HIV proteins. HIV gene expression. So, for example, this bar where there's the most SSU72 and the most TAT has the most HIV gene expression. And so following that pattern, they looked at what would happen if they mutated SSU72. And what they found was that in TAT negative cells, like they expected, there was very little HIV transcription. But in TAT positive cells, mutated SSU72 had significantly less HIV transcription than in normal wild type SSU72 cells, which helped them to find that mutating SSU72 may be helpful in affecting the function of HIV in cells. So then the researchers looked at what would happen if they knocked out SSU72 and how that would affect TAT's function and HIV gene expression. And what they found was that, once again, TAT negative cells, very little HIV transcription. But in TAT positive cells, knocked out SSU72 had significantly less HIV gene expression than in control cells with nothing knocked out. It's important because it finds that knocking out SSU72 is effective in fighting, not fighting, but affecting HIV transcription and how much of it is expressed in an affected cell. Over on this graph, they compared SSU72 to other CTD phosphatases to see if it was unique in its function. And so these four are just other CTD phosphatases, the same thing that SSU72 is, and they found that in these CTD phosphatases, there was not an effect on HIV gene expression. However, with SSU72, there was, once again, a significant effect. So it established more clearly that there was a unique link between TAT function and SSU72 function. So obviously there's a lot to go from here, but really, what is the next step in HIV research? How can this be effective in humans, and how does this compare? How does this compare with the current picture of HIV treatments? Sorry. What do you guys think? And do you have any questions about?
Do you know if researchers can use this newly found pathway to treat HIV?
Well, I mean, that's kind of the question is, it appears that it would be effective because they found that it does decrease HIV gene expression. Yeah.
What are the other two bars in the graph there?
These, this is another protein that TAT recruits to the RNAP2 that is part of a complex that moves along transcription. This one I'm not entirely sure. I believe it's another one. Yeah?
What's the y-axis there? I can't read it at all.
Fold induction, it's how much gene expression.
So that's fold. Right.
Yeah.
Yes.
What's the difference between graphs A and B?
A is comparing, well, it's mostly just looking at SSU72 knockdown, and this is more comparing it to other CTD phosphatases, other proteins that have essentially the same function as SSU72, but different in specifics, and finding that there's...
Did they explain why only SSU72 is able to increase transcription?
Well, that was sort of the idea, was that because of its specific function in affecting S5P, mostly was that it moved along transcription in the process of promotion, so TAT was able to use SSU72 to expedite transcription, move it forward, whereas, I guess, the specifics of these CTD phosphatases, which phosphate groups they removed, weren't as effective, or didn't do anything, weren't related. TAT didn't interact. Any other questions?
phones on them?
Yes.
A computer, a phone? Raise your hand if you've liked the Project 80 page. For everyone that doesn't or hasn't liked it, please do that, because there is where you're going to start getting all of your updates on what's happening in IRT, Project 80, Journal Club, and all the other sciences in the schools. And if you're looking to